All right, I think we can start now. Would you be happy to start now, and Professor Ellington?
That'd be fine. Sure.
Okay. Let me just. Cool. So, hello everyone, and welcome to our third seminar of the term. Today's talk will be given by Professor Andrew Ellington, Professor of Biochemistry at the University of Texas, Austin. Professor Ellington's lab does work on a wide range of synthetic biology topics, from novel genetic codes to point-of-care diagnostics. Today's talk will cover his work on the use of yeast synthetic biology to study complex systems such as the brain. It is my pleasure to welcome Professor Andrew Ellington.
All right. Well, thanks so much for the invitation. Again, in these trying times, I would much rather be over there talking to you in person, and hopefully I'll manage to get through this and leave enough time that we can discuss. But people should feel free to drop in or out as they need to. So let's see. There we go. So just to get it all straight at the outset, I'm not a neurobiologist, and that's fine. But for those of you who tuned in to hear about neurons, this may be problematic. That said, we work with some excellent neurobiology collaborators, and actually we're excited to find more should any of you be interested. This deficit in my background should not particularly worry you, as really synthetic biology is sort of about the ability to move between systems. I actually remember in the early days of operating systems, giving you an idea of how old I am, I mean computer operating systems, there was this dream that one day we'd be able to move without any care between different operating systems, which is pretty much where we are now. And synthetic biology is now moving towards the same sort of interoperability for organisms. So ignorance is not quite bliss, but it's enabling. We'll be discussing a variety of relevant high throughput screening and directed evolution methods, and I will endeavor to give you, some insights into how they can be compared and contrasted. Okay, so the rough structure of the talk will be an introduction to chemogenetics, and then we'll talk about making ion channels work as synthetic biology parts, making GPCRs work as parts, and finally, depending upon if I decide we have enough time left, talking about making drugs that can work on ion channels or GPCRs. Okay, so the reason we chose to think about chemogenetics was because really the basis for much of neurobiology is the synapse, and either the release of ions or neurotransmitters at the synapse or the modulation of that via basically chemical effectors as well as sort of electronic effectors up and down the neural pathways. In order to do this, we need to think about where chemistry actually comes in, and that leads us to receptors and drugs, as it does for much of biology. You essentially have proteins, small molecules or peptides or other proteins interact with those proteins, modulate their function, and thereby lead to some sort of output. In this particular case, the output is in a neuron, either relatively fast action potentials via ion channels or relatively slow changes in the capabilities of a neuron via G protein coupled receptors. And that's why we're going to talk about both of these. They operate at different timescales, but being able to control both these timescales should prove useful. Now, one of the most exciting things, I think, in neurobiology these days is optogenetics, the ability to use light to change the function of receptors and thereby downstream change neural function. And so I think we'll see if this movie works, but I just find this to be totally cool. Oh, there we go. So here's a mouse, and we turn a laser on and we excite particular engineered neural receptors, and it begins to eat. And it's being told, as it were, to eat by the light. And then when it turns off again, it says, well, I guess I'm through eating for now. That I'll just sniff at the food. So I'm not going to watch the whole thing here, but that to me is just an indication of how this notion that we can go from chemistry and receptors upwards towards neural pathways and behaviors is really the right way to go. Now, while optogenetics is really, really, really cool, the fact is that it has issues, right? Unless you want to wear, sort of a optical probe in your brain, it's highly invasive. You can't access necessarily deep tissue, it's certainly in a human, and the light can potentially damage cells. So we're instead thinking about how we can generate orthogonal control over neural systems via chemogenetics, being basically using drugs to control particular receptors or control particular neurons. Now, again, what this is going to require is something that is sort of the same old shtick for my lab. This is things we've done again and again. It's controlling what proteins can interact with what compounds. It's really protein engineering, but it's protein engineering for a particularly difficult class of molecules, receptors, and we've made some advances there, which I'll talk about in just a second. So, for example, if we can make the effector for an ion channel like P2X2 not be AMP, which is its normal effector, or ATP, which is normal effector, but rather AMP PNP, which is a non-hydrolyzable analog, that will give us some way to control that particular receptor however we wish. So this is in general what we're going to try and do. We're going to try and create orthogonal control pathways via ion channels and GPCRs. Okay, now we do not in any way, shape, or form come into this in a vacuum. And so I think you probably invited me, because most of what I'm about to show you we haven't even published, but I think you invited me because we wrote, this nice little review on synthetic neurobiology, and hopefully we are doing justice to some of the other methods out there which try and control interactions between receptors and chemicals. For example, DREADS and DART, which are two very nice methods. They mostly rely either upon high throughput screening in the case of DREADS, or upon sort of changing the geography of tethering in the case of DART. Now, we think that we can get better control over orthogonality by actually controlling the drug compound interaction and by being able to do higher throughput screens and selections. Nonetheless, this is where the state of the art is, and what you're about to see is things that we haven't published, but certainly we're part of, or we'd like to think we're going to be part of this community, and I wouldn't want to imply otherwise. All right, so as is the case for, I'm pretty sure, all of you, all the work in my lab is not done by me, all right? I mean, I am the first to admit that I am a glorified figurehead. This particular set of work that I'm going to talk about is largely due to Elizabeth Gardner, who's an extraordinary graduate student who took on this project several years ago at her own behest. My lab did not used to work in this area, but Liz really wanted to work here. And so what she decided to do was first try and figure out, along with a neurobiology collaborator, Boris Zemmelman, who's her co-advisor, whether or not we could make an ion channel work in a system where we would have greater control over engineering. And neural systems are difficult to work with, but this is an interesting candidate for doing this because, as we saw on a previous slide, it's activated by ATP. We have a way to control it, potentially orthogonally. It's expressed in the peripheral nervous system, so again, we can potentially put it places that it will have interesting control features. And it would potentially make a much better tool than the currently used channel rhodopsin. The optogenetic system has better conductance, so it gives really a great spike upon activation. It desensitizes in some cases slowly, and there are a variety of phenotypes. So there along the bottom are several different P2X receptors that have been tested in human mammalian cells, and their various action potentials in those cells. And you can see there's a variety of spike characteristics. But over there in the lower left, if you compare this with channel rhodopsin, we would have roughly the same conductance, but we would have potentially, for example, for P2X2, a much slower desensitization, I should say, which might give us, longer term control over action potentials. Okay, but what Liz decided to do several years ago was see whether she could make this work in yeast. And I probably don't need to tell you, but I will belabor the point that yeast is a great eukaryotic system for doing protein engineering and for manipulating proteins in general. It's easy to culture. You can do large library sizes, much larger than if we were going to do DARTs or DREADS. Genetic modifications are relatively straightforward. Even chromosomal modifications are easy. And it's amenable, of course, to screening and selection strategies. But Liz encountered one huge problem when she decided to create her own hurdles to jump over, and that is that no human ion channels had previously been functionally expressed in yeast. So this was Liz's own characterization of what she needed to do: express the ion channel, find a candidate for channel engineering, characterize function, and then finally move on to screening and potentially selection. And as you'll see, she's almost through with this painful path. We're just in the final stages, as you'll see, but we're not quite completely through this train yet. So here's what she first did. She needed some way to determine whether or not an ion channel had a function in yeast. So if it is in fact letting in calcium molecules in the presence of, for example, its cognate allosteric ligand, ATP, how could we even tell? Liz initially used a quarin, which produces a very nice luminescent signal in the presence of calcium. And she can read that easily on a plate reader, and thereby we could potentially use light to monitor something akin to the ion flux across the yeast membrane. Rather than doing an action potential in human cells, this would be sort of light-mediated changes due to the influx of calcium, or sorry, calcium-mediated changes leading to light. All right, so this was the scheme. And what was incredibly surprising, especially to me, was that this worked. So we saw that there were 7 receptors, and there are 6 of them over there on the left, P2X1, 3, 4, 5, 6, and 7. And you can see out of P2X4 and 7, we get a little bit of reaction. So that's really great. And actually, if we were to go back here just briefly, if you scan there along the bottom, you can see that 4 and 7 were 2 of the human receptors that gave a sort of broadish signal. 1 and 3 gave very narrow spikes, and 5 gave very little signal at all. So it's perhaps not surprising that 4 and 7 are giving a broadish sort of response here in yeast. But over on the far right, what was the true surprise was that P2X2 just blew the top out of our assay. So it also, and I could step back again, but I won't, gave a broad signal similar to 4 and 7, but 4 and 7 don't even register on the RLU axis that P2X2 works on. You can see there. So, you know, the height of the peak for 4 and 7 is around 2,000. The height of the peak for P2X2 is around 600,000. So it's pretty remarkable in terms of how well it works in the yeast membrane. But this also tells us that not everything's going to work exactly like human cells. And so that makes us both happy and suspicious at the same time. Will we truly be able to use yeast as a model system to essentially study neural proteins, ion channels? We actually call these yeastons as a sort of, you know, yeast that are neurons, but is that really going to be true? Nonetheless, Liz is 2 clicks along in her pathway, and while P2X2 is a standout, again, we are curious whether it's going to really work the same way it does in human cells. So next will be to characterize ion channel function in yeast. And there, what Liz has been doing is she's been basically trying to see whether or not you can get a gated response in response to ligands. And so we previously talked about how ATP is the normal ligand, and lo and behold, we do get gating based upon ATP, and it actually pretty closely matches what is expected for the human ion channel. That is, if it were expressed in human cells. She also tested other known agonists and antagonists of the channel, and they also worked similarly. So the ion channel implanted in yeast not only works, but is working, at least from a chemogenetics point of view, in a similar way to what is seen in human cells. She then tried to see from the receptor side of things whether this all made sense. And so, like any good molecular biologist, she first killed it. She went to the literature and found mutations, in this case, a double mutation from lysine to cysteine that was known to knock out activity due to basically mutating a key pocket based on the zebrafish structure shown there with 2 red arrows. And when she does this, it flatlines. That's the orange line there. And so, yes, in yeast, if we introduce mutations known to kill the channel, it kills the channel. All right, that's a necessary demonstration. It's probably not as impressive as, oh, it responds to the same effectors as the human channel. But then what she did was she looked in the literature to find population-based mutations, alleles in the human population known to cause a disease state, in this case, inherited deafness. And the actual activities of some of these mutations are known. And R290K, the human allele arginine to lysine at position 290, is known to be partially functional, and even though it causes deafness. And lo and behold, in our assays, it's partially functional. That's there in the middle of that bar graph. Whereas other mutations that from the human population known to be dead are dead in the yeast assay. So Liz has done a really good job now of showing that she's 3/4 of the way through the path she set out for herself. She expressed ion channels. She found some that could be potentially engineered that had functions similar to those in a human cell. So now, where do we go from there? Now, this is where we begin to not just demonstrate that we can do this, but provide some level of control. What she's done is mutagenize, again, the binding pocket for the purinogenic ligand, ATP, and we're going to now try and find variants of the ion channel that can respond not to ATP, but to AMP, PNP, which is a non-hydrolyzable analog. And thus, again, you can imagine if we were to express a mutant channel in neural cells eventually and be able to introduce AMPNP into the bloodstream of an animal, that would be a great advantage over trying to introduce ATP, which breaks down relatively quickly in the bloodstream of an animal. So she first wanted to mutagenize this NFR motif. And just something I will tell all of you, graduate students in general love animations. I typically take out most animations in talks. And of course, Liz helped make this talk and she was amazing and awesome, but everybody loves putting in animations. I usually take them out, but this one I think will make sense. So she first identified those mutations and then she did site saturation mutagenesis at those two positions in the binding pocket. She eventually wants to do more portions of the binding pocket, but initially it was just that tyrosine and that asparagine that were mutagenized. And then she just basically plated out the library. Those are individual yeast, each one of which has a different library member, which leads us to be able to do a screen by picking library members into individual plates and then putting them in the transformation from calcium to light assay that was previously shown. And so when Liz does this, she can then pick the mutations that look like they yielded light and then sequence them. And again, this is just a screen, it's not a selection, but it's great to see that it works. So when Liz did this, she was able to go from a flatlined dead version to back to wild type. So that's what's shown there in yellow. That was a picked mutant. And when she sequenced it, it had snapped back to being asparagine, AAT, shown there in the sequence of the clone and along the bottom in terms of the original clone. And while if you're really a skeptical scientist, you may say, wait a minute, maybe she just got a few wild types mixed in with her library. In fact, she's also got different clones that have different codons. And so we're pretty sure that the positive variants, the active variants we get back from this screen are actually, from the library and not just from wild type contamination. So here's where she's going next. She's actually got a really good collaboration with a microfluidics lab to greatly increase the throughput of the assay. Because right now, she's picking colonies into plates at the hundreds, and we really want to go, to the 10,000s or hundreds of thousands for larger libraries to look more broadly at what mutations might lead to the ability to bind AMP, PNP. And while we've been using a Aquarin in the past, there are limitations on that, and she's recently switched to engineering calcium-dependent transcription factors, where the response isn't as fast, but nonetheless, for the purposes of screening, we may not care. And letting in calcium in the presence of ATP, or eventually we hope AMP, PNP, will lead to basically calcineurin regulating transcription. Okay, so the really key things there, which I'll emphasize at the end of the talk as well, is we have now the ability to potentially work with ion channels in yeast cells. That's never been seen before, and that could potentially be really, really interesting and important. And I'm sure many of you who are synthetic biologists can imagine fun experiments of trying to get yeast to talk to one another in terms of using ions or other neurotransmitters. We're not necessarily going that route. We're not sort of doing the sender-receiver system things that synthetic biologists often like to do. But it's pretty exciting, and we want to work now to having a part that is responsive to AMP, PNP, or other ligands, and then reimplanting that in a neural cell and eventually in a transgenic animal. So going back to the very first of my talk where I said, bummer, I'm not a neurobiologist. Nonetheless, I think that this will be a good way to launch ourselves back into the brain. Okay, let's talk about GPCRs. So the great thing about GPCRs is while they are on a completely separate time frame, they are really interesting molecules in their own rights. 30% of drug targets are GPCRs, and there's hundreds and hundreds of these things in humans that regulate a wide variety of aspects of biology, not just neurobiology. So being able to do this is just generally important. Being able to have chemogenetic control via screens and selections over GPCRs would be very worthwhile. But I want to re-point out that other folks have been out there, sort of plowing this field with dreads and darts and other approaches. Now, we would like to do this in yeast because that allows us to isolate a GPCR pathway relative to the literally insane complexity of GPCR signaling in humans. So there on the left is, a typical set of interactions of GPCRs in humans. And again, to show my age, in the early days of trying to study transcription factors, when most of the systems were bacterial, they made sense. Lambda repressor made sense. Lac repressor made sense. They were turning on or off specific operands. And then came along eukaryotic cells, where suddenly transcription was just a mess. And I had colleagues at the time who just used to use the acronym TIS in eukaryotic cells, which stood for transcription is stupid, because it made no sense, because we didn't really understand complex networks back then the way systems biologists do now. We were used to thinking of sort of one signal in, one signal out sort of stuff. So that's where we'd like to be again for GPCRs, in a simplified system where we can control and understand signaling is what we're trying to get to. We're literally trying to reduce the complexity on the left over there to the right. Now, there are very good methods by, and what that also allows us to do once we gain that control is we could potentially put it in neural cells and via promoter specific expression, express particular GPCRs in only a particular subclass of neurons. And that's already well worked out. So while we're going to talk about transcription in a couple of ways, either transcription signaling via GPCRs, there's also transcription of GPCRs in particular neural cells that will make this a good approach. Okay, but in terms of the simplification, taking the wild type yeast pathway, which operates through the yeast pheromone receptor, ST2, and then converting that literally almost the only GPCR in yeast to working as a human GPCR involves inserting the human GPCR, shown there on the right in blue, and making a chimeric G alpha, where that chimeric G alpha has enough of the human factor, usually at its amino terminus, in order to interact with the G alpha can now interact with the rest of the human GPCR. So this chimeric humanized pathway, again, operates in a completely insulated orthogonal way. It's pretty much the only thing in yeast you're signaling with. But now, instead of signaling hormone, it's going to signal whatever, one hopes, the human GPCR initially signaled in human cells. So it's basically extracting enough of the human system so that we don't have this complex mess shown on the previous slide. I can't even get back to the previous slide. But instead, being able to, there it is. I just moved, okay. So rather than that mess on the left, we again just extract out one piece of it and put it in the yeast there on the right. Okay, great. And again, that's been done before. I should point out that humanization of GPCRs is sort of a big business at this point. In fact, there are entire libraries of these. Nonetheless, we wanted to make particular variants of human GPCRs work that we thought would be important for human neurobiology. And in particular, what we wanted to do was then take those human GPCRs and make them into particular variants that would be activated by the compounds we wanted them to be activated by. It's exactly the same thing we just talked about a little while ago for ion channels, except now with GPCRs. Now, we call this concept overall high affinity validated orthogonally couples, or HAVOCs. We want couples between particular drugs, which may not even be found in a human, just like we were talking about AMP, PNP, the non-hydrolyzable ATP analog, wouldn't be found in a human or a transgenic animal. And then couple that with our specific receptor, which would have great orthogonality, great specificity for the new drug. So you can see where we're coming in terms of a chemogenetic approach to control. One of the first ones we were interested in was CB2, the cannabinoid receptor. Now, there are two cannabinoid receptors, CB1 and CB2, but we got fortunate in that when Liz cloned the CB2 receptor into yeast, we were able to get activation of transcription using the chimeric G alpha I3 factor in yeast with 2AG, and which is not shown, but it's an endogenous cannabinoid, and beta-caryophyllene, which is the compound shown there on the left. So we get activation of the receptor by the compounds that normally activate it. I'm now going to digress just a minute. Okay, so at this point, keep in mind that we've got a chemogenetic approach for ion channels. We're going to make a chemogenetic approach for GPCRs, but the next three slides are going to be a digression because I'm sort of excited about them. We want to eventually make not just channels for chemogenetic approaches, but drugs themselves. And so we're sort of excited about getting into engineering cannabis. So the lab is just beginning to take its first tiny steps into being able to engineer cannabis to produce novel cannabinoids. Now, this is harder than it sounds because no one really has done much in the way of GMO cannabis so far, or GMO hemp. And so this is a prophetic, we're not yet regulated license to do this, but this is what we're moving towards slowly with American regulators doing. And we have an excellent collaborator in this regard, Alan Lloyd. We previously worked on engineering dandelions. And that work also isn't out yet, but I'm not going to expostulate too much on it. But we think we're going to be able to use some of the same novel methods that we've used to transform dandelions to be able to transform cannabis, and thereby do things like make a non psychoactive cannabis. If we knock out the THCA gene that's involved in the actual production of THC, the active compound in marijuana, we can get CBD producing hemp that should be able to redirect their carbon in a metabolic engineering way to high levels. But more than that, we can begin to look at, there are literally, sort of something like 200 different cannabinoids in your average hemp or cannabis plant. And so being able to at will redirect production to particular of these cannabinoids, many of which are grossly understudied, but look as though they'll have really interesting outcomes in terms of treating pain or actually just producing a variety of interesting neurological effects, we think will be just a gold mine. It'll just be a lot of fun to do this. But again, this is in the service of, to back up just a bit, finding new drugs that can then either interact with CB2, with variants of CB2. You can imagine if we can redirect the production of cannabinoids towards a particular cannabinoid, we can then make that cannabinoid and look and see if it interacts with CB2 and eventually CB1, or even creating entirely new cannabinoids. Now, why would we want to do this in the plant? Why am I engineering the plant? The plant's really good at production. I mean, you have chemists who certainly can make synthetic cannabinoids. You can produce these biochemically in cell culture, but the plant is just ridiculously good at making cannabinoids. And so we think that this is actually how you would eventually want to make it for a market as well. And we're always interested in potentially translating our endeavors. Okay, so we have CB2 working in yeast. We have on the side an entire effort, hopefully, in producing novel cannabinoids. Now, how are we going to do screens or selections? And so I'm going to segue here just a little. Previously, with the ion channels, you saw that we did high throughput screens. We turned calcium into light via an ion channel. Here, we're going to rely upon transcription, where we're going to actually tether a peptide adjacent to not the cannabinoid receptor. So I'm pulling a bait and switch a little bit on you here. To make the system work out, we're doing a proof of principle with a peptide binding GPCR and tethering a peptide adjacent to it. So we have so-called autocrine activation, right? We have self-activation of a peptide produced in yeast and sensing of that peptide by the human expressed in yeast, GPCR, and downstream transcription. Okay, Liz has also built out a really nice yeast toolkit to basically put in peptide production with appropriate tags for expression and various GPCRs with various export signals to get them up into the periplasmic space of yeast. And again, we're just going to read this out downstream by turning on transcription and making some GFP. And we're going to now, on the slide after this one, report on peptide-based autocrine activation as a fold change relative to a non-cognate peptide. So the right peptide should make the yeast green, i.e. somatostatin receptor and the SST14 peptide, and the wrong peptide, i.e. the alpha factor, which the yeast normally produces, shouldn't turn on that human receptor. And yay, it works. So actually, Liz didn't just do the somatostatin receptor, she did a wide variety of known peptide receptors, and you can see some of them don't work very well. So there's HSKOR. That's the human opioid receptor. And you can see there in the middle, it doesn't work very well, right? It's not getting turned on either at pH 5.8, a yeast-like pH, or pH 7.1. One of the things we had to learn was we needed to actually buffer the yeast so that the human receptor, which of course grew up in human cells, would work better. Whereas if we let yeast be yeast and it reduces pH, lo and behold, some of the receptors don't work. But under any conditions, that beta endorphin receptor, that opioid receptor, doesn't work. But the ones in red work quite well, including the somatostatin receptor, CXCR1, and the vasopressin receptor. So we got this to work to a certain extent, and other folks have shown this as well, but we've expanded significantly the number of peptide receptors we can work with in yeast. And now again, we're proofing this ultimately for things like the cannabinoid receptor, but a cannabinoid is obviously not to a first approximation a peptide, although we could of course try and find peptide mimics for cannabinoids. One of the reasons things didn't work was because, again, yeasts aren't human. And so we took a page from our colleague Ed Marcotte, who works down the hall, and we introduced, and this actually worked by Colleen Mulvihill, who's a joint student between my lab and Ed's lab, portions of the human cholesterol biosynthetic pathway into yeast. So yeast normally have ergosterol, but we said, why don't you make cholesterol? And shown there on the right, a lot of white space, I know, but this is the mu, not the kappa, but the mu opioid receptor working in a human cholesterol engineered strain. And so what's really cool here is that we're slowly but surely turning yeast into human cells. And the mu opioid receptor is just, again, a gold mine of a target in terms of doing neurobiology. And so we're pleased to see that here, where we can potentially eventually do a peptide, it's not a cannabinoid, but we can potentially do endogenous peptides that activate opioid receptors, endorphins, that we could potentially do some engineering. But this is just all sort of scattershot telling you about the difficulties we've encountered trying to get GPCR engineering and selection going. Here's some more of that difficulty. What we'd eventually like to do is something like this, where we had a GPCR library that we transform into yeast, and different library members would either be activated or not activated by either a wild type peptide or a peptide that we were able to put up on the surface. You can imagine peptide libraries versus GPCR libraries. Now, the way we typically do selections is called CPR, not PCR. CPR stands for compartmentalized partnered replication. And so I'll come back to this slide in just a second, but I'm going to once again slightly digress, and say, this is compartmentalized partnered replication in bacteria. And what it is we just drive, rather than GFP for an optical screen, the production of Taq polymerase. A good partner gene that drives the production of Taq polymerase can then be amplified once a bacteria is emulsified. And it doesn't matter what the partner gene is. For example, Adam Meyer in my lab made T7 RNA polymerase variants in bacteria that drove the production of Taq polymerase, and in the context of an emulsion, allowed the amplification of those variant T7 RNA polymerase genes. And this just shows 6 different RNA polymerase variants that Adam was eventually able to make work based upon selection in E. coli, right? So this is just a generalized emulsion-based method for selection. But now we're coming back to yeast. We're going to do the emulsification. We're going to drive the production of Taq polymerase. But now the variant partner won't be an RNA polymerase, it'll be a GPCR, or it will be a peptide partner. And so upstream of all the transcription will be a variant that either will or won't lead to the production of Taq polymerase, that either will or won't then amplify the gene that led to its production. Now this is non-trivial. This is really non-trivial. All of our previous attempts to make CPR, compartmentalized partnered replication, work have been in bacterial cells. But a few years ago, Jimmy Gallagher in my lab tried to begin to make this work in yeast. And so while these methods are 3 years old at this point, more than 3 years old at this point, we haven't really until recently begun to apply them in yeast, because it's just taken a long time to get to a system where we can go from a receptor to transcription to production of a polymerase to self-amplification. So Jimmy first had to go through and optimize emulsion conditions. He had to optimize protocols for yeast lysis. And eventually he got self-amplification of the polymerase gene itself. So this is so-called compartmentalized self-replication, which was, give all credit where credit is due, developed by Phil Holliger at Cambridge, who's just an amazing scientist. I hope many of you get a chance to meet him during your careers. And so Phil has been able to do self-evolution of polymerases. We use this technique to instead modify other aspects of expression, T7 RNA polymerase for transcription, transcription factors themselves, and in this case, really moving upstream to receptors and potentially peptides or other compounds that could activate those receptors. Okay, and so this is where we are. This is literally, as of, I think, a couple of weeks ago, where we are, where we can get somatostatin activation by an anchored peptide, and we don't produce green, we instead produce a Taq polymerase that can self-amplify in the context of emulsion the gene for the receptor that led to its activation. So all the pieces are working. And now what we're doing is working on making libraries of receptors, or making libraries of peptides, or making libraries of peptides plus receptors to basically mutate and optimize the function of the somatostatin receptor. Now, that's not going to be so important for neurobiology, but GPCRs are GPCRs, and we want to work our way back to either the mu opioid receptor, which looks like it's now working, or cannabinoid receptors, which are obviously working. And now then, instead of having self-production of a peptide and anchoring of it, you can imagine the instantiation of the metabolic pathway. People already got yeast that make opioids and cannabinoids. And so we will have the pathway for the production of opioids or cannabinoids, leading to the production of a molecule that activates the receptor for an opioid or a cannabinoid, which leads to transcription, which leads to production of a polymerase, a thermostable polymerase, in the context of an emulsion, rather than self-optimization of the receptor or self-optimization of a peptide that activates the receptor. You could have self-optimization of a pathway that leads to the production of a compound. I hope that made sense. If not, you can ask me questions about it. Okay, let me check on time here. We got, nominally 20 minutes left. I actually have, a number of slides left, and I think unless anybody objects, and anybody can speak up and object, I'm going to go through the last part here. But it's quite different than, not quite different, but somewhat different than the first two parts. But just to recap, I gave my explanation for why we want to do chemogenetics, how we have now made ion channels work for the first time in yeast as parts, and how we can begin to do screening, how we and others have made GPCRs work as parts, and how we're working towards selections, not just screens, but real selections where we literally self-amplify genes that help produce a polymerase. And finally, I'm going to go to a third different system for making new transcription factors, which will help make drugs that can work on ion channels or GPCRs. So again, this is a large-scale effort in my lab to do metabolic engineering in a smarter way. So if nobody objects, I'm going to head on into this, but I want to make sure we have enough time to talk with either. Okay, so how can you actually make natural products? I gabbled about, boy, wouldn't it be nice to be able to make cannabinoids or opioids or whatnot, and maybe we can do self-optimization of pathways in our super cool, hopefully, yeast that both make and sense a given compound. But the fact is that just making those compounds is difficult. And what you really need to do in order to scale up natural product production when you're working with a heterologous pathway, and it almost doesn't matter whether it's a heterologous pathway in bacteria or yeast or plants or whatever, is you need to be able to sense the molecules that you're making. And that's going to be true, especially if you ever move towards high throughput production. I'm already pretty confident that we're not going to make cannabinoids in culture, although you could. We're going to make them in the plant, but other compounds you may want to make in culture, and there again, you're going to need sensors. So we have new mechanisms by which we're going to make new types of sensors and regulators for individual compounds that we hope will feed back on pathways that produce those compounds. So this is, again, going to be a generalized method, but we're eventually going to cant it towards, as you'll see, being able to improve opioid production. All right, so this is all work by Simon Dolsnitz, who sits right next to Liz Gardner, and they talk a lot, and they have their own ideas. But Simon's going to now be working not in yeast, but in bacteria. And in bacteria, he's going to try and basically turn TET-R transcription factor members. So these are the same TET-Rs that are tetracycline regulated and can be either on or off, including in eukaryotic systems. I'm sure many of you are familiar with TET-R-based transcriptional control in a variety of systems. But we want to make it so, oops, is this gonna, oh, it's gonna, oh, gracious. All right, so this is on it. I forgot this was on a timer. It doesn't matter. I'll just flip it back when I need to. But we're basically going to make TET-R families members do what we want them to. This is good because TET-R family members already have large ligand binding cavities, good flexibility, amino acid conformations all over the place, and they already recognize structurally diverse ligands. But what Simon really wants to do is he wants to improve opioid production, alkaloid production, in a variety of ways, but he needs individual sensors to improve individual steps in the process or individual end products. And so what he cleverly did, and Simon is very clever, Simon and Liz are just amazing, is he took a bunch of different alkaloids and then he looked to see whether any of them could interact with the TET-R family member RAM-R. And shown here is just initial results of a screen showing that THP, tetrahydropapaverine, can interact with RAM-R. The other ones not so much. And keep that in mind. The other ones really not so much. But Simon decided RAM-R was a good starting point for making libraries to identify RAM-R family members that would interact with these different alkaloids. And so he's going to go from a semi-specific family member, RAM-R, to, as you'll see, highly specific transcription factors for sensors and transcription factors for different alkaloids. All right, so this is a fairly complex set of screens and selections, and I'm probably just going to buzz through it here in the interest of time, but it's a two-step process by which he screens for active receptors in the presence of a compound using GFP as his readout. And then he selects against constitutively active repressors in a negative selection. So this is a screen. You got to pick the green colonies. This is a selection. Things that are constitutively active that don't wait for their alkaloid, just plain die. And then this also allows him later down the line to do a counter selection so that if it's not reacting only with the compound of interest, it dies. So this is all summarized here, where Simon will take his library, and it doesn't actually have to even be a very big library. He transforms it, and then he first selects for things that are constitutively active that die, and then only takes things that haven't been activated constitutively. And then he screens for things that are only activated by the alkaloid of interest, and then he further validates those and eventually ends up sequencing mutants. This looks really, when I first saw this, I was like, God, this is kludgy, Simon. We were working on all these crazy emulsion-based systems where we could screen or select through, hundreds of thousands of different variants at a time. And here we're doing, 10,000-ish sort of these things. And, we have to do sequencing and so forth. But in fact, Simon proved me wrong, as most of my students do. And he quickly was able to make a RAM-R variant that was responsive to noscapine over only 4 rounds of selection with targeted libraries. And so that's, goes from the wild type, no reactivity, to round 1, 2, 3, 4, really starts popping up around 3. He did this for every alkaloid he tried. He was able to get individual receptors that were reactive to individual alkaloids. And he was able to, over time, because of his counter selection, go from the sort of mess of a diagram you see there on the left, where you have some cross-reactivity, even after the first round of selection, to the diagram on the far right, where it is almost perfectly specific for a given alkaloid. We actually were curious about how in the world he did this, so we collaborated with crystallographers, and we got crystal structures of all the best variants with their variant alkaloids. And we're not disclosing here, because we're in the process of intellectual property protection, exactly what mutations these are. But Simon found that there was a wide variety of ways that you could accommodate new alkaloids in what was originally a binding pocket that was only vaguely specific, the original RAM-R receptor. Okay, so I'm almost done. What I wanted to show you in this last part of the talk, and we'll still have a little bit of time to talk, is that directed evolution can be used to generate transcription factors and biosensors for a wide variety of ligands. And while in the middle portion of the talk, I showed you emulsion-based selections CPR, compartmentalized partnered replication, can lead to good sensors, the ability to readily screen for signal. Now we're back to signaling just like we are or back to screening just like we are in the first part of the talk, which Simon called proxies, gives researchers more control. And we go back and forth with this argument in my lab often. Should we screen? Should we select? Which is better? More numbers, better control. And we probably will go back to taking some of the concepts we've learned here for possible applications to yeast-based human receptors or even transcription factors in yeast that could be responsive to compounds, again, like opioids. Semi-specific receptors can evolve to be highly specific based on binding site changes that can post facto at least be rationalized. And now we're finally to where we sort of are, or hope to be soon with cannabis, making biosensors and transcription factors that for cells can be used for strain optimization. If you want to make a particular alkaloid, you can sense it and then have feedback loops between that sensing and the improvement of the pathway to make that alkaloid. Okay, and we're finally done. That's my lab there, sort of in the middle. Special thanks to Liz Gardner, who you heard about in the first part, Simon, who you heard about in the last part, Shaunak Carr, who's just a generally good synthetic biologist, supports many of these efforts, Jimmy Galahar, who I mentioned, who was a graduate student in my lab and now runs a lab adjacent to mine for the Army. We have actually an Army research lab, and Jimmy is really the king of high throughput biology. He's remarkable. The Zhang lab helped us with crystallography, especially Nate Burkholder. And we have support from a variety of agencies, but mostly I thank you for your attention, and hopefully we have time now to discuss, including any of that wasn't clear. But to recap, we can make ion channels work in yeast for the very first time. We can, essentially have a screen for their production and use. We can make GPCRs of interest work in yeast, human GPCRs. We have a selection scheme that is just about to work after many years. And finally, we're optimizing transcription factors so that we can optimize metabolic pathways for the production of metabolites that have neurobiological interest. As I said at the very, very, very beginning, none of that's neurobiology, but hopefully it's going to be fun. All right, with that, I'm looking at the chat. And other than that, there's nothing in chat for me to answer, so I'm happy to just answer your questions.
Right. Thank you, Professor Ellington. That was a fantastic talk. If you guys want to ask questions, just turn on your videos and we'll call you one by one. So,
may I start off in that case? I have thank you very much for the talk. I have a question with regards to the GPCR autoactivation, where you modify the pH to be more human-friendly as opposed to more
yeast-friendly. Yes.
Yeast-friendly. Can this be used in some way by as a sort of a switching, or is this just an interesting little phenomena?
That is a very interesting question, Raymond. I had not thought of that. That's a really cool question. Again, for those of you who are curious about sender-receiver systems or communication systems, we're trying to essentially, and Jeff Bookey did a nice job of this a few years ago, but we're trying to put more peptide-responsive GPCRs into yeast so that we can create networks of yeast that make specific peptides or respond to specific peptides. But Raymond, you've come up with a new twist on that. If in making such networks of systems that make and sense peptides, you also have the option of yeast that say reduce pH in their local environments, you could have an additional switch on top of that. So that would be pretty cool. We're not so much doing that right now, but I think it's a really interesting idea. And again, for many of you, I'm sure synthetic biology that addresses communication systems is really interesting. And complex communication systems are now becoming possible with these yeast-based systems, but nobody's really exploited that yet in sort of a everybody talks to everybody way. Maybe that's too long an answer, but I think I got at your question.
Yeah, thanks. Another possible question is,
sure.
With regards to a talk, like we already asked you whether you'd be comfortable with this being recorded, but you did mention that there was some stuff that was cylinder
Right,
underwork?
but that's why there's no labels on that.
Okay.
Right, I've given that portion of the talk before, and it's pre-protected. There's nothing in, I mean, these are definitely some unpublished results in many cases, but I haven't said anything that is going to harm any intellectual property endeavors. And in general, while I'm very, very, very interested in intellectual property and commercial translation, that comes secondary to my primary mission, which is to be an academic and to put out my students' work. And so again, the real take-home message from this work is that, you know, I'm very proud of Liz and Simon, and I would encourage any of you who are interested to contact me, because, you know, I'm always looking for interesting folks to work with, including if including if any of you are real neurobiologists or want to take some of these systems into real neurobiological applications.
If there's any other questions.
Hi, Professor Andrew. Hi, I'm Betty, and thank you for your talk. And I just have a quick question about the Taq polymerase induction. So you mentioned that after inducing the activity of the GPCR, which will lead to the Taq polymerase production, and I wasn't quite sure about how do you actually screen for the Taq polymerase.
Right. So that is an excellent question. Do I say Yunjiao? Is that right?
Yeah, I'm Betty. Just come Betty.
Close enough. Okay. So what happens, Yunjiao, is in an individual cell. Here, I will use my hands. This is typically what I do when I'm teaching. You know, in an individual cell, will produce the Taq polymerase. So GPCR will be activated, produce the Taq polymerase. That cell is in turn in an emulsion ball. Okay. So just like when you send samples for sequencing and do, say, emulsion PCR, you just thermal cycle the bubble and the cell blows up. That allows primers in the bubble to get access to the gene for the GPCR that led to the production of the Taq polymerase. So I know that all sounds really complex, but it actually makes sense. The GPCR variant turned on the expression of the polymerase, and now when the cell blows up inside the bubble, the polymerase and the primers can amplify the GPCR variant that led to the production of the polymerase, right? So now all you have to do is imagine that bubble next to a million other bubbles, and you see that if you're a dead receptor, then you don't make any Taq polymerase. If you're a great receptor, you make a lot of Taq polymerase, and you self-amplify the gene that led to making a lot of Taq polymerase. Does that make more sense?
Yes, thank you.
Okay, I hope it did, Junjiao. We actually have several papers out on compartmentalized partnered replication, but not in yeast. That is actually a major innovation in our lab, at least, that's taken years to finally get to the point where we can now apply it to things like GPCRs and ion channels and so forth.
Yeah, and of course, I'm going to write down. Thank you.
Thank you.
Is there any more questions?
Hi, thank you for the excellent talk. I would like to ask whether there is a limit to how many humanized GPCRs you can have in a yeast population, or whether you've tried to basically combine a few to make a more efficient system.
That's a great question, Petra. There are two ways to phrase that. How many GPCRs can you have on an individual yeast cell, or how many could you have in a population of yeast where each one had a different GPCR? Which one of those do you mean?
Possibly both.
Good, that's what you should mean. Excellent. So I think that Jeff Bookey, when he published on the peptide receptors, had something like 8 different peptide producing, peptide sensing yeast at a time. We think we could probably take just about double that number now. That is, that you'd have 16 squared possible interactions between peptide producing and peptide sensing yeast. And keep in mind that some of those could be the same yeast. You could produce peptide A with sensor B in one yeast, and then in another yeast you'd have sensor A and produce peptide C or D or E or F or whatever. So I think you really can at this point create complex networks. But your more interesting question is, in fact, well, that's nice. Could you produce more than one GPCR in the same yeast? And the answer is there's nothing that said you couldn't, but the signaling pathway, which also pretty much goes through ST2's mechanisms, would not be orthogonal. And so what you'd have to do would be duplicate and diverge the communication pathways down to transcription. Now, I don't immediately remember anyone who's done that, although I bet someone has. But if you wanted to put more than one GPCR, you'd need separate communication pathways. Somebody who has done a really good job of that in a different context is a guy named Jeff Tabor at Rice University. And in bacteria, not yeast, he's begun to use a variety of two-component systems for sensing, but then has orthogonalized, has made separate pathways for interaction with downstream transcription factors. So Jeff really can put up multiple different two-component systems on the surface of a bacterial cell and then communicate separately to genes in the bacterial chromosome. But right now, I think it would be pretty much one yeast, one GPCR. So that's going to be a limitation. But in terms of number of different GPCRs that could talk to one another across a population of yeast, I think we could now probably get up to around 16 squared. Does that sort of answer your question, Petra?
Yeah, absolutely. That's very exciting. Thank you.
Cool. And again, these complex systems are now out there, and I would encourage you to at least think about, for those of you who are also not just neurobiologists, but systems biologists, I think that making these synthetic communication systems, complex synthetic communication systems, is a real issue, right? It's like, well, how do I really communicate across a bunch of cells? Can you essentially create multicellularity with multiple different, you could just call them hormones, they're peptides, but just call them hormones, all communicating separately, and what does that mean in terms of the system? So I think there's a lot of play in these systems. We're just down in the grunt level developing the parts necessary to create complex circuitry. I often find that students like you are way better at thinking at complex circuitry than I am. I'm an old school biochemist. I make molecules, and I do like working with people like you and my colleagues who work in complex systems. But I feel like if we have a good part, then it will almost certainly lead to an interesting system. But I'm not necessarily the best person to imagine that interesting system. I would say you guys probably are. And I would love to talk with you about that into the future.
Thank you for the answer again.
No problem. Okay, well, if there are no more questions, and I'm happy to take any more questions, all of you are more than welcome to contact me individually or independently at any point in your careers. This is the funny thing, is you will see as you move along that, we bump into each other all the time. I still bump into people I was in graduate school with. So it's surprising how the community narrows and how it's the same folks. And, we should just keep talking as peers, as scientists. And I'm really excited that I got to give this very long distance seminar, but I really wish I was there going to a pub with you. I really would rather that be the case. So hopefully maybe in the future we can meet up someplace.
Yeah.
I actually have a question.
Sure.
Thank you again, as everyone else has said. This is really interesting. Have there been efforts where people have tried to use yeast and E. coli with these signaling systems?
That is an awesome question, Olivia. Yes, these co-culture systems are quite interesting. One of the reasons that yeast are yeast is because over the years, as it were, they've learned to get rid of bacteria, right? So I'm excited because I get to go back to my graduate student years. I worked on yeast alcohol dehydrogenase when I was roughly your age. And the thing about yeast is they puke, so to speak, alcohol because they're trying to kill all the bacteria around them, right? They basically clean out their competitors and then have this big pool of alcohol that they take back up and metabolize. So they essentially, have this interesting ecological strategy. That said, it is entirely possible to find bacteria that just thumb their nose at the ethanol, or to make the yeast make less ethanol, or not quite as low a pH. And so I think these co-culture methods are very interesting. They're often exploited in bioprocessing and sometimes even in real fermentations. And again, synthetic biologists have the great advantage of not caring what organism is what. And potentially engineering those systems into the future? So it's a really good question. It overcomes evolutionary barriers set up by the organisms themselves, but in ways that we can readily imagine. Not so much pH, not so much ethanol. Bacteria, take it up a notch, deal with a little more ethanol, right? These are all engineering questions totally within our ken. So that's a great insight.
Wow, that's really cool. Yeah.
All right. Awesome. Thank you very much, Professor Ellington. And I hope to see the rest of you very soon as well.
Yeah, really, thank you very much. You guys made my day. We'll talk to you soon. Bye.
Talk to you soon.
Thanks again.
Thank you. Bye.